Okay, I think we are ready. Good afternoon, everyone, and welcome to the College of Optical Sciences Colloquium series. It's my great pleasure to introduce our speaker today, Dr. Anita Koshy, who is a doctor of medicine, so she's a real doctor, a lot like me, right? So she's an assistant professor in the Department of Neurology and Department of Immunobiology here. She's local, so that's great to have her here. So Anita studied biology at Stanford, and then she also went to school at Duke University Medical School, right? And then she did a bunch of residencies in, neuroinfectious diseases at the University of Washington, and then University of California, San Francisco, and then Stanford University. So a lot of good places that she has been. And then she did also a postdoctoral fellowship in the lab of John Boothroyd, so where she studied cells in the brain that have interacted with the parasite Toxoplasma Gondii. Hopefully I said it right. Okay. So now, after a bunch of education, a lot of research, she's now good enough to come to U of A to set up her own group in 2012. And she has been studying the molecular underpinning of the brain toxoplasma interaction. So I think it will be a very interesting talk for us to listen to, a different one. But let's join me to welcome Anita.
I don't know if this is on. Is this on?
Yeah.
All right, how about now? Yeah? I'm usually plenty loud, so if I'm not loud enough, let me know. Let's see. Okay, so thank you for the great introduction. It's really fun to be here. This is sort of a challenge for me, which is how to get people who don't think about parasites in the brain to be excited about parasites in the brain. And as we go through this, we'll talk about why I think optical sciences and biology are really well suited to help each other in understanding these issues. So the basic outline is I'll talk about the background. What is toxo? What does it do? I'll talk about the impact of imaging on toxoplasma biology. So I'll talk about it both from the general standpoint and then very specifically on where I think it helps with the CNS. I'll talk about the Toxoplasma-Cre system, which is a system that I set up as a postdoc and that my lab relies on heavily here, and it's very imaging intensive. And then we'll talk about just some future directions and the collaborations that we have already and why I think there's some good opportunities, especially in terms of long-term in vivo imaging. Okay, Toxoplasma Gondii. Who in the audience besides Dana Zach has heard about what Toxoplasma Gondii is? Okay, so very few people, a couple people. Okay, so it's a eukaryotic parasite. Just means it has a nucleus. It's a protozoan. Means it's one cell. It's a relative of malaria. To give it some context, so there's a whole sexual life cycle over on this side that only happens in the cat. It's not relevant to this talk, so we're not going to go over it. But one of the most important things about toxo is that it has a huge intermediate host range. So this is very atypical for parasites. So it can infect everything that's basically warm-blooded. And there's someone who's made a huge career out of documenting all the natural infections that occur in toxo, so from birds to humans. And very importantly, in both humans and rodents are naturally infected. So we work primarily in mice, and that allows us to have a natural model to study things. So the way that this works is in most intermediate hosts you ingest, so you eat your toxo through contaminated food or water. It then goes from a dormant state into a fast-growing state. It spreads all over the body of the animal, and the immune system kicks in, clears it out, except for a couple places. So that where it doesn't clear it out, it depends on the animal. In humans and rodents, your main reservoir is your brain. Okay, and then in humans, we also know it persists in the cardiac muscle and the skeletal muscle. But the importance of this brain is that we think that once you have toxo, you have it for the rest of your life, and the major area where you carry this parasite is in your brain. So a rough estimate to bring you some context is about 1/8 to 1/3 of the world is infected with toxo. So 10 to 20% of the U.S. So 40, 50 people in this room, 5 people in this room are infected. Okay, and we think you have it for the rest of your life. Hanging out in your brain, which sounds sort of interesting. But we think in general that if you have an immune system As long as it works, your CT, your MRI, whatever you want to scan, your PET scan would look normal. There's no way I can detect someone who's toxopositive versus toxo-negative by any sort of standard imaging or interaction. But since we can't cure anyone, so there are no drugs that will cure you of this disease, there are a couple instances where toxo becomes very clinically important. So if you get this infection when you're a fetus, if your mom gets infected while she's carrying you, and particularly in the first trimester, this can lead to some very devastating consequences. So this CT should look essentially like that one. It obviously doesn't. The other time that it becomes very important is in patients who get immunosuppressed. So AIDS patients are the major time when we saw a huge outbreak, but now toxo has been associated with bone marrow transplant and with basically all the new immunosuppressives. Okay, there are at least a couple cases in each of the anti-TNF antibodies and various other monoclonal antibodies. So my interest in toxo is twofold. Is one, it would be nice to definitively cure this patient of toxo, or if we knew someone was getting a bone marrow transplant, to be able to treat their toxo definitively prior to them getting that bone marrow transplant. And I think we do that by understanding how toxo persists in the brain. The flip side of that is that toxo is very efficient at persisting in the brain. And so it seems to have co-evolved with the mammalian brain, at least the rodent and human brain. And so that means it is evading the brain immune response, and the brain definitely has an immune response. And so if we can understand how toxo evades that brain immune response, then potentially we can harness that for our own utility. And the reason that becomes important is because the brain immune response, or neuroinflammation, has now been implicated in everything from multiple sclerosis, which is the classic autoimmune disease of the brain, to traumatic brain injury, to Alzheimer's disease. Okay, so if we can understand how toxo is co-evolved with the brain immune response, potentially that gives us a really powerful tool to manipulate the brain immune response in our own way. So how can imaging and optics help us understand toxoplasma biology? Well, there are a lot of things parasites have to do to ultimately get into the brain. So they have to cross barriers, and optics can help us there, and I'll show you a couple examples of that. It can help us understand what occurs at a cellular level at the host-parasite interface. And then it can help us figure out where, sorry, that's the host-parasite. You guys are one behind my slide. Where the parasite is located in the CNS. And that's important because it looks like there's some regional preferences, and that might speak to how different parts of the brain have different immune responses. So just a little introduction to the brain and the cells of the brain. So everyone pretty much knows about neurons. Those are the big cells. They carry the electrical current. The oligodendrocyte, that's the myelin, or the insulation around the nerve. So these are the cells that get affected in MS, where you get or any demyelinating disease. Then you have your astrocytes. These used to be considered just pretty much support cells for the neuron. They're now recognized to play a huge role in everything from setting up neurodevelopment and synaptogenesis, so the connections you form, to being a critical part of the innate immune system. And then there are the microglia, which are the tissue-resident macrophages of the brain. So basically the brain's immune cell that lives there. Okay? And for toxo, the things that have been implicated are the microglia, the astrocyte, and the neurons. And we know that the neuron is where the parasite persists primarily. But the microglia and the astrocyte have both been implicated in helping control toxoplasma. So the brain is not a good place to get an infection, as you might imagine. And so the brain is a very fine system, a variety of fine systems set up. And one of the bigger things that it has set up to keep things out is the blood-brain barrier. And this is a very specialized barrier that allows the brain's extracellular milieu, as well as the cells that come in and out, to be finely regulated. Now, it used to be thought that immune cells didn't come into the brain, that you had to have a breach in your blood-brain barrier for immune cells to come in. That's been definitively disproven. But what happens is immune cells come in, and specifically T cells come in, and if they don't find their correct antigen, then they track out. So if you look at a brain and you slice it and look for immune cells, if it's uninfected or there's nothing going on, you're not going to find a whole lot of T cells. Okay? But if you give an animal a strong enough systemic response so that all the organs are sort of on alert, you will see that T cells now track into the brain. And a lot of that work has been nicely done with in vivo imaging. And so what separates the brain's immune system, or the blood-brain barrier parts of it, is that in most organs, when you get down to the capillary level, so the very fine level of blood vessels, they're fenestrated, meaning that they don't join up perfectly. And that's where you get exchange of material. So that's where oxygen and carbon dioxide go in and out, things like that. And in the brain, those are tight junctions. So there's no way for molecules just to diffuse in and out very easily. And that's of a particular size. So these tight junctions are a big issue. In addition, you have these parasites that surround the capillaries. And then just beyond the capillaries in the post-venual state where the immune system comes in, it doesn't have it here, you'll have these astrocyte end feet and this basement membrane, and usually there are some immune cells that sit in there. So there are a lot of things that the parasite has to get through to be able to come through into the brain. And so people have not directly studied this. I'll show you one study where people are beginning to study it. They've done it mostly in vitro. There's some issues there. And the major ways that people think that toxo gets into the brain is either by being carried by an immune cell, so what's classically called the Trojan horse method. Some viruses do this, but so the immune cell gets called into the brain because there's a systemic immune response going on, and if it's infected, it can carry toxo in. The other way that people think about potentially toxo getting in is that it actually directly invades. So that can it invade into this endothelial cell and then come through, or can it come through between the junctions? One of the things that argues that toxo should be able to come through those junctions is that when you ingest your toxo, it becomes a free-living parasite in your gut for just a little bit, and there are a bunch of tight junctions the toxo has to get through again. And so if it can get through the gut barrier, it can probably get through the blood-brain barrier on its own. But that is harder to, there's less evidence for this one. So the first thing I'm going to show you is a nice movie about a couple of movies. So this is, first movie this is out of Melissa Ledowen's lab. This is published work. And these are in vitro systems where they're looking at epithelial cells. And the first movie is just a monocyte, so just an immune cell crossing the barrier, crossing into doing transepithelial migration, or TEM. So that immune cell is going to go, and you see how it flips under? Okay, there's a lot of little molecules involved in that. So then they wanted to say, so what happens? There's some indirect evidence that toxo makes monocytes move faster when they're infected and do further migration. So then they decided they wanted to look at, well, what happens when you have an infected cell? So here it goes. You can see it going around. I'm going to stop in just a minute if I can. Okay. And so what happens here is you see that immune cell pops under. This green stuff, these green things are parasites. And what you can see, and I'll keep playing it in just a minute, is this immune cell, as it's trying to go through that layer, is getting stuck because the parasite's migrated to the back of that cell. And now it's stuck on it because it's a hard thing that it has to get through. And no one had conceived of that until Melissa imaged this, right? It was just like, oh, I'm sure the immune cell just gets through just like that. Well, turns out it's actually a little more difficult than we thought. But it will be able to do it. But it's really fascinating, right? So it has to exert a fair amount of force to actually get through that cell. There's another group, Chris Hunter. This is all in vitro, or what we'd call in vitro. Biochemists wouldn't call this in vitro. And this is a group that does 2-photon imaging, live imaging. So just to put it out there, we would love to do this here. So we'd love to do it in a much more long-term way. So they thin the skull of the mouse, and then they image. And so they can image for a couple hours. And these blood vessels are green because they express GFP off an endovascular marker, so Tie2. And right here, this is a set of parasites. So they intraperitoneally infected this mouse, and then I can't remember how long this is. It's probably 2 weeks or 3 weeks later. They thinned the skull and went and looked at the vessels. And you can see right here, that's a group of parasites. That'll become a little more obvious in just a minute, but the color of that parasite is red. And so they just watch what happened. So this is Christoph Conrad from the Hunter lab. This is not published. Okay, so you can see those cells, it ruptures, or the parasite egresses. Most of the parasites get swept up into the blood, so they can go off to other distant tissue. But a couple of them stick around, and so you can imagine that if they can do that, maybe they can rupture back the other end into where the parasite is and do a cycle where that's how they're getting in. Okay, so it's not clear how these things get in. No one has imaged immune cells infected immune cells crossing the blood-brain barrier, and that's because that is a relatively rare event. They're hard to catch, and so people haven't done it. Okay, so what is my lab study? So my lab study is the question of how does toxoplasma persist in the CNS? And we're particularly interested in what does the brain bring to it. So this is an EM done by a friend of mine, Carolina Cafaro, of a mouse that was infected, and then 3 weeks later, its brain was taken to EM. And these are each individual parasites, and they're in a big cyst. This whole thing is called a cyst, and you can see, if you look carefully, there's this little bit of plasma membrane that goes around here. So this parasite, all these parasites are in a cell, which makes sense. Toxoplasma is an obligate intracellular parasite. But what's different than, say, something like a virus that can integrate into the genome or that can go very dormant episomally, Toxo's not doing that. This is a huge inclusion in this cell. Okay? So our hypothesis is that there's communication going on between this host cell and this parasite. And the parasite's highly manipulating this host cell, like most obligate intracellular microbes do, and it's doing that to allow nutrient acquisition and evasion of the immune response. Okay, so that host cell becomes very important in trying to understand what's going on to the host cell. So what other evidence do we have for that? Well, we happen to know that during acute infection, which is what most people study because you can do that in fibroblasts, the toxo injects a bunch of effector proteins. And those effector proteins go and highly manipulate the host cells. So if you take host cells and you take uninfected cells and you do microarrays, about 40% of the transcriptome will have changed by being infected. Okay, so that's a huge amount. And some of these changes have been mapped to these effector proteins, but a very small number. So we know that toxo manipulates host cells with these effector proteins like most intracellular microbes. And so our theory is that that happens in the brain as well. So what we'd like to get to is being able to understand in vivo and at the level of a single cell what's going on in this parasite-brain interaction. Okay, so we want to get to this single cell because I've just told you that I think the host cell is where everything happens. That's how toxo mediates its responses and its global responses. In addition, we want to work in vivo because from that first slide of the brain, it's a really complicated organ. You've got a bunch of different cell types. You've got neurons in different regions of the brain can be different, and even astrocytes in different regions of the brain can be different. So trying to mimic that in vitro at this point I don't think is very effectively done. So how are we going to get to be able to work at the single cell level, identify these host cells, and work in vivo? And so we turned to something called site-specific recombination, and we adapted that for host-parasite interactions. And site-specific recombination is just the idea that you can temporally and spatially modify genes. Okay, and there are a bunch of systems to do this. The system we chose to work with is the Cre recombinase LOXP system. Cre recombinase is an enzyme from a bacteriophage, and it recognizes in bacteria these very canonical sites called LOXP sites. And people recognize that, and then they said, hey, we can use this to our advantage. So huge numbers of biologists use the Cre LOXP system to specifically turn things on and off in very organ and cell-specific manners. And the way it works and everything that I'm going to show you is you have a promoter and you have a GFP cassette. And this GFP cassette is not expressed because it's got a stop that is flanked by 2 LOXP sites. So it's a flox stop reporter system. So when you have Cre, you need, in theory, 4 molecules of Cre recombinase, so it's a very sensitive system. When you have Cre come in and cause Cre-mediated recombination, now your promoter is going to be next to your GFP, and it'll read through. So how are we going to sort of weaponize our parasite to do this? Well, we took advantage of those effector proteins, and we basically just fused Cre to the back of one of those effector proteins. So now when those effector proteins get injected into the host cell, this Cre-mediated recombination should occur, and our host cell should turn green. Okay, and all of our parasites, we generally have them express a red fluorescent protein so we can see them easily. So this worked very well in vitro. So this is a control parasite. They're red, but the cell hasn't turned green. It doesn't carry Cre. This is the one that carries Cre, and you can see that this cell has turned green. And we know in both of these situations, it's just a single parasite that's come in. So they've come in and then they've divided in both of these situations. And we know that because there's only a single parasitophorous vacuole. So all those parasites are grouped together. And that's really important that a single parasite could trigger this because that's what we think happens in vivo. So then, of course, we really want to go into the CNS, and we could show that, in fact, we could get Cre-mediated recombination. And so you can see this is a mouse that was infected in its abdomen or intraperitoneally, and then 21 days later had its brain taken out and we sectioned it. So all these green cells have interacted with the parasite. And there are actually far more green cells than we expected. I'll show you this, just sorry. This is off a different section, but it's a nice section. So you can see here's our parasite surrounded by green. Here's another green cell. These are neurons. This one actually colocalizes with one of these. This is just an antibody stain, and these are astrocytes. But what was interesting when we did this, and we had some hint that this might happen, is that there are a bunch of green cells. So in a highly infected mouse, you have about 3,000 to 10,000 cysts or infected cells at this time point. Okay? This is one section of about 300 sections that you generate from a brain. So if you counted up these green cells and then you multiplied it by 300, you'd get on the order of 150,000 to 300,000 cells. That left us a lot of cells that were not accounted for. And so we knew some of this happened, sorry, one more, which is that in culture, right, in vitro, we could see these cells that were infected. So there's parasite, that one's turned green. Next to it's a green cell. Now if I blew this up, you'd see these two are connected. So that one was very easy to explain. That one just divided after Cre-mediated recombination. But then we'd find these cells like this one. This one actually has a parasite in it. This one, where there's no parasite in there, and there's no parasite around it, and there's no green cell next to it. So they clearly didn't happen from dividing and then moving over a lot of room, because that's not what happens with these types of cells. And so that led us to recognize that there were some important things about this Toxoplasma-Cre system, which is that it permanently marks host cells regardless of infection status. And that's a new way of thinking of things, because prior to the system, we defined host cells by who was infected. Right? That was the only way we could define host cells. Are you a host cell or are you infected with toxo? Okay, now this system says you don't have to be infected with the parasite, right? You just have to be injected with this protein. And so what that allows us to do is that we can recognize the infected host cell, but we can also recognize the host cell that's had the parasite come in and then through cell intrinsic mechanisms cleared toxo. And we know this happens in toxo in a variety of cell types. And we can recognize the cell that's been injected by toxo and never invaded. Now, we can sort these two out in vitro. We can't sort them out in vivo. Okay, so these two cells will look the same to us in vivo, because we just don't have good ways of separating those. And then any of these three, if they then divide post-Cre-mediated recombination, will also be green. And what's important about that is, who cares if you could find that I can turn some cells green, but in published work that I'm not going to talk about, we could show that these cells, all these green cells, both in vitro and in vivo, looked very similar to infected cells, to actively infected cells. They were manipulated akin to that. And so what that means is those 150,000 to 300,000 cells in the brain are all probably manipulated in a way that we hadn't conceived of previously. So that really changes your dynamics. This also let us do something which was sort of fun for us, which is test our current model of CNS-Toxoplasmosis. So the current model, based on in vitro and in vivo data, says that the parasite comes into the brain, and then it infects your astrocyte, your microglia, and your neuron. And then using cell-intrinsic mechanisms, your astrocyte and your microglia can clear the parasite. Your neuron is immunologically inert, which is how neurons are often thought of, and so it remains infected. And that is consistent with the in vivo data that we know that cysts primarily encyst in neurons. But you could never test that model because you couldn't find those cells that were previously infected, right? So now we can do that. So this is what we would expect to see. So now in our system, parasites come in, turn a bunch of cells green. Whoever it goes into should turn green. And so now what we should see is that we should see a bunch of microglia and astrocytes that aren't infected but are green. Okay, that's what this model would predict. But surprisingly, that's not what we got. So we get tons of green cells, and we can get green cells that are microglia. So that we're not finding. But what we couldn't find were astrocytes. So the majority, this one is an image of 3 weeks post-infection. And what we do is we then take that section and we stain it for neuronal markers and astrocytic markers, and then we just co-localize. And these three are shown there, but these guys would also co-localize with neuronal markers. So we know those are neurons. They morphologically look like neurons. They co-localize with neuronal markers, so they're neurons. And so that was really surprising, because this is not what the model says. The model says neurons interact with parasites because They just happen to be there, and then they just can't clear them. Okay? And astrocytes are supposed to be a really important cell in all of this. And the importance of that is that there's been a huge focus in vitro of direct astrocyte-parasite interactions. And this data suggests that that may not be relevant. But we have to go prove that first. So we counted up these cells, and at 5 days, which is the first time we looked, there's no green cells, and there are very few parasites in the brain. At about a week and a half, you can see that there are lots of green astrocytes. So these are percentages of green cells. There are lots of green neurons, very few astrocytes, and then a lot of immune cells. This is not surprising. And that's true at 3 weeks. And then by this time, these unidentified cells are actually not immune cells, but are more often neurons, or morphologically look like neurons that aren't staining anymore. And it's probably because they're dying. So if we just look at the astrocytes and the neurons, you can see that the neurons predominate. Okay? So these are the neurons, these are the astrocytes. Now, astrocytes, people used to say that neurons were outnumbered 10 to 1 in the brain. If you really look at astrocytes and neurons, it's about a 1-to-1 ratio in the rodent cortex. So if it were just by chance the toxo is going to go into whatever cell it hits, you should get a 50-50 ratio. Okay? We clearly don't see that. We can do that with a second strain of toxo, and we can get the same answer. Okay? So this isn't some, because we're using some very specialized strain of toxo. So then, of course, like any scientist, we wanted to know why do parasites predominantly interact with neurons? And one of the big things that comes up is interferon gamma. So interferon gamma is the key cytokine for being able to keep your parasite in control. If you don't have interferon gamma, you're not going to control your parasite. And one of the big things about that is that interferon gamma has a huge number of effects on many cells. And so the way we went about answering this is we interferon gamma depleted mice. And then we wanted to also test very specifically if the major way that interferon gamma is known in astrocytes to kill intracellular parasites, this IRG system, if that was involved. And we did that by creating parasites that are resistant to this system. And we can do that because we can play between neuroscience and parasite biology. Okay? And then the next question we want to know is it the location where the parasites come in? So maybe that explains it. And we're going to get around that by actually intracranially injecting parasites. And then the last question, is it the neuron surface area? And we'll get back to that. Okay, so here's what happens if you interferon gamma deplete parasites, right? So this is the major mediator of parasite persistence. And all of a sudden, you get a bunch more green cells. And you can show, and this is just a totally uncontrolled toxo infection. So you can see this is a control mouse, got an antibody that wouldn't deplete interferon gamma. And you can get the GFP cells in the control on any given slice about 15. It's 122. In the interferon gamma depleted mouse. The cyst count, so our best way of estimating how many parasites are, 8 versus 100 versus 50. So we were talking about a very uncontrolled infection. And in that situation, we can show that you can get about 30% astrocytes of those green cells. So interferon gamma seemed to be playing a role. We can do it this way too, where we're just looking at interferon gamma, astrocytes and neurons. But what we couldn't distinguish here is it just you have an uncontrolled, completely haywire infection? Okay, so then we want to go more specifically and look at these IRGs. So IRGs so mice have a huge number of IRGs. Humans have very few IRGs, but they have an equivalent, another set of TPases that function like IRGs. And toxo, as in any sort of arms race, has its own proteins that block these IRGs. So the IRGs load onto the parasite and destroy the parasite. Toxo has a protein that's able to block that ability to load. But only certain strains of toxo are able to do that because they carry these two functional proteins. And that's ROP18 is a major mediator, so this is the one that it's a kinase, it phosphorylates the IRGs, and that keeps them from being able to load. Okay, and the other thing you need is ROP5, and a very specific allele. And so one of our parasite lines has ROP5, but it doesn't express ROP18, so it is very IRG sensitive. So we can then just express ROP18 in our parasites. We can engineer our parasites to do that. And then we can ask, what happens if we infect them with the ROP18 who are now IRG resistant, right? Now, if the IRG theory is correct, if that's the major mediator of astrocyte killing of these cells, then we should see a bunch of green astrocytes here, because now they can't kill that parasite. And it's not what we see again. So what we see is that the resistant parasite looks just like the sensitive parasite. So it's not that we're missing astrocytes that are green because they're killing the parasite and then dying themselves. There's some evidence that they didn't die after they deploy their IRGs. So that's what it looks like if you just look at astrocytes and neurons. And then we wanted to say, okay, well, let's really force this system. Can we force astrocytes to interact with parasites? So we took uninfected mice, and we actually injected the parasite into the brain. Totally unnatural, but it allows us to be able to control where we put it. Okay, so we're going to put it in the neocortex where we know the astrocyte to neuron ratio is 1:1. Okay, so now there's no interferon gamma around, so there shouldn't be anything to block, because you don't express interferon gamma at high levels unless you're infected. There shouldn't be anything to block the parasite from being able to interact with the astrocyte. So this is what it looks like. So this is what happens if you inject a PBS mouse. You can see the level of injection, and here's what happens if you inject parasites. There is red in here. It's just hard to see, but you can see there's a bunch of green cells. So again, now we're at a 1:1 ratio. We should see if it's just the toxo hits whatever it sees and goes into those cells, then we should get a 50-50 ratio, and we don't see that. Again, we still see astrocytes just don't interact with parasites very often. So this neuron-astrocyte interaction is very common. And there seems to be some sort of preference. And it's not clearly a preference because we think part of what may happen is that the parasite just interacts with neurons because neurons are so much bigger. So numerically, they're equivalent to astrocytes, but neurons are huge cells. And this movie hopefully will help illustrate that. So this is a 200-micron thick section that has been optically cleared and then imaged by traditional confocal microscopy. And then Imaris, which is a commercially available software that's very expensive, has been used to map out all the areas that are green, right? So you can see that the cyst is right here. These are all our little red parasites. But look at how much surface area that parasite had to interact with, right? It's a huge and massive expansion compared to an astrocyte, which are much smaller from a surface area standpoint. Just to show another movie, because I actually love these movies. So here are 3 cells. Hard to tell who's infected. There's your parasites. But again, doing these thick sections where you can reconstruct everything. At some point here, it'll stop spinning. And you'll see that this connection, when it turns, maybe, is very clearly connected to this cell body right here. So you can see that little thing. But look at this network, right? If you were a parasite migrating here, right, that's a pretty juicy section to be able to choose any of those. So we don't know if it's simply that it's just a surface area issue, or are there actual specific factors that cause toxo to want to interact with neurons more than astrocytes. Okay, so our new model of toxoplasma looks like this. So the parasite comes in, it specifically interacts with neurons, and I've shown these 3 neurons as being infected, but I can't prove that, okay, because all I know is that they're green, so at the very least they've been injected with parasite protein. And then at the end of it all, we end up with very few neurons that are infected and lots of these green neurons that have been injected. And so either they're just getting injected, or what's more exciting for us is the potential that they are being infected and then are clearing that parasite, because that would be a whole new paradigm. Neurons shouldn't be able to do that. So my take-home points are that imaging makes a huge difference in understanding the biology, right? Everything I've shown you today is about how imaging really helps us understand things. And there are things that just imaging, that simple assays like Western blots just don't get at. There are a lot of subtleties there. Our own system, the Toxoplasma-Cre system, is very imaging reliant. And it's helped us, has allowed us to rethink our Toxoplasma-CNS paradigm. And one of the nice things about this is with any new tool that you build, you'll usually learn something that wasn't known before, because it just opens up your ability to think about something. So astrocytes may mostly work through indirect mechanisms of controlling toxoplasma. We know they're important, but they probably don't actually do a lot of intracellular killing of parasites because they probably don't get infected. And the corollary of that, the flip of that, is that this neuron-parasite interaction is very important. Okay, so we have to stop thinking potentially of neurons as immunologically inert. They have immune systems. They're probably capable of clearing parasites, and we just have to figure out how. And so optic biology collaborations will hopefully fuel further discoveries. So the future directions that we're doing already is we'd like to image a whole intact brain. So right now we can image a 200 micron section, and we can image a very small section of that. Okay, so we would like, there are newer clearing methods, and we can clear the brain, and now we'd like to be able to image the whole brain, or at least big chunks of the brain, and see how these patchwork of green cells, do they connect, or is there something that associates with them? What can you learn from imaging in a much more three-dimensional way? And part of the problem with that is just the sheer amount of data that takes. That takes a lot of time, and it generates a lot of data. And so we are already working on this. And then with Khan and Nasser, we're working on imaging infected brain slices without antibodies. And that's hugely important and of great importance to most of neuroscience, which is that if I take even a 200 micron thick brain section, right, to get antibodies through that brain section is very hard. There are only so many fluorescent proteins you can express and then image. In addition, there's now evidence that doing a ton of fluorescent protein expression is not necessarily normal or good or healthy for those cells in which you are expressing them. Okay, so if we can image them, and this is optics, you people understand this, third harmonics, fourth harmonics, whatever it is, so that we can come up with cell signaling things that don't rely on antibodies, that's a huge advantage to us, right? So instead of showing you a green cell, I can show you a green cell with my astrocytes layered in there, with my microglia layered in there, right? And we can begin to understand that cellular dynamic in a way that you couldn't before. And then I'll make one more pitch again. I'd love to perform in vivo two-photon imaging here. Okay, so we would love to be able to watch how does the parasite get into the brain, but even more interesting to us would be how do those green cells get involved, right? How do they can we follow a parasite as it either injects, then gets invaded into a neuron and then gets cleared, or that it injects and never invades into that neuron? Okay, so those are things we would love to do. And with that, I think I'm done. So that's my lab. And our funding sources, and that's it. So I'll take questions.
Okay, question, guys. Any question from the audience? Just go ahead.
You mentioned thinning the skull damage.